Welcome to Trial Site News Podcast Series. Today we will be discussing drug side effects, which can range from mild to fatal. Now, they can occur when taking a drug as prescribed or as a result of incorrect dosages, drug interactions, or off-label usage. Now, according to some reports, adverse drug reactions are responsible for about 2 million U.S. hospitalizations per year, according to the Department of Health and Human Services, and even occurred during about 10 to 20% of the time during hospitalizations, as reported by Merck Manuals. And so today, to talk about this and more, we'd like to introduce a postdoctoral fellow at Harvard Medical School, Robert Ietswaart. So Robert, welcome.
Thank you very much for the invitation, Adrian. I'm very excited to share our work on this Trial Site Network podcast today.
Now, Robert, could you share more about your background? I'd like our audience to get to know you a little bit more.
Yeah. Indeed, I was born and raised in the Netherlands, and I studied mathematics and physics at Leiden University. And there's a little story to that. I wanted to be a scientist ever since I was a child. My parents were not scientists, but a personal role model was my uncle, who pursued a PhD in biochemistry when that research field was only just emerging in the 1970s. Later, my uncle became a professor at Leiden University Medical Center. But my interest in biomedicine early on was also quantitative in nature. I wanted to mathematically model cellular processes. And to be able to do that, I would need a quantitative skill set. So I decided to study maths and physics because at the time there was no such thing as bioinformatics courses. So my studies prepared me well to pursue a PhD in computational biology in the United Kingdom. And now I'm a postdoctoral fellow in the lab of Professor Sterling Churchman at Harvard Medical School in the genetics department. And these quantitative skills continue to serve me excellently for machine learning and biomedical research, such as the work we will be talking about today.
Now, can you share with us how the team got together? We know there was some sort of hackathon organized by the drug company Novartis. Is this true?
Absolutely. Novartis organized a hackathon back in the summer of 2018. Novartis has been one of the first pharmaceutical companies to realize the potential of utilizing big data and machine learning to improve their business and research. So they organized this quantitative sciences academia industry hackathon, where graduate students and postdoctoral researchers in academia would learn about the research and development process at a big pharma company like Novartis. First, we learned about pharmacokinetics and pharmacodynamics modeling in drug development from the hackathon organizers Dr. Miriam Tram and Andrew Stein. In the second week of the hackathon, this was devoted to research projects in teams. So representatives from different divisions within Novartis pitched their research problems, and the students then chose to join a project team based on their interest. Dr. Laszlo Urban, the global head of preclinical secondary pharmacology from the preclinical safety group at Novartis, told us about their work on predicting drug side effects during the drug discovery process. So a total of 7 students joined the team, and Dr. Urban with a colleague mentored us throughout that week. It was fun, and an intense hackathon. By the end of the week, we had developed an initial version of our machine learning model. The hackathon concluded with a symposium within Novartis where each team would present their results. And after the hackathon, 5 of us remained committed to further develop the study with the aim to publish our work together with Dr. Urban.
So what university and companies came together, and can you share the makeup of the team that developed the AI algorithm?
Yeah, so the hackathon participants were really coming from academic and research institutions across the US. Besides Dr. Urban and his colleague, Dr. Duncan Smith from Novartis, our team consisted of Seda Arad. She was a computational biology postdoc at the Jackson Lab in Connecticut. Amanda Chan, a PhD candidate in tissue engineering at MIT. We also had Saman Faramand. He was a PhD candidate in bioinformatics at the University of Massachusetts. And Boom Joon Kim, he was a PhD candidate at Northeastern University. And since the hackathon 2 years ago, it's been a while, right? Like some of us have moved to different positions. For instance, Dr. Arad is now working at Pfizer as a computational toxicologist.
Wow. So we can certainly make the case that drug side effects are incredibly costly to society and to people's health. Can you share on whose idea this was and if there was any commercial tie-ins?
Yeah, for pharmaceutical companies like Novartis, clinical trials are a huge cost with high risk of failure. Overall, it costs on average over $2 billion to bring a new drug to the market, and only around 5 to 10% of candidate drugs that enter clinical trials get eventually approved. And the first phase, like the phase 1 trial, is simply to test for drug safety. If a candidate drug causes too severe adverse events in patients, the trials will be terminated. So any information that can help predict whether a drug candidate is likely to cause side effects already at the preclinical stage, that is before it is tested in human trials, that will be very valuable. Most importantly, it would limit the risks for patients undergoing the trials, but it also reduces the opportunity costs to a pharma company like Novartis. If we could prioritize safer compounds to enter clinical trials, this could in effect reduce the developmental cost and potentially increase the overall rate of testing new medicines. This ultimately improves the health of the public. Dr. Urban has spent most of his career on drug safety research from within Novartis. During the hackathon, he presented to us his research problem of using in vitro target-based pharmacology to predict drug side effects. In vitro target-based pharmacology characterizes the binding properties of drugs to proteins of interest with in vitro experiments in the lab. Then it was really a team effort where we brainstormed to see what quantitative approaches, such as machine learning, we could apply to solve this problem.
Now, Robert, we've taken a keen interest here at Trial Site News in artificial intelligence and related technologies such as machine learning and natural language processing. Before getting into the material of the study, could you provide a brief primer on what AI is and what are high-level differences between machine learning, NLP, and other forms of AI?
Certainly, yeah. Artificial intelligence, or AI, often raises our imagination, right? Because of what we see in sci-fi movies. But AI is really a broad umbrella term. AI covers every non-human system, program, machine, or robot that can solve problems intelligently. Machine learning and natural language processing, or NLP, they are subdisciplines of AI. In particular, machine learning basically means computer algorithms that improve their performance. In particular, machine learning basically means computer algorithms that improve their performance automatically through experience. Mathematical models that utilize training data as their experience enable learning a certain task, for instance, automated language translation. Then, after this training stage, the model can make predictions on test cases that the model has not experienced before. The ideal model is one that requires very little training data, but can make accurate predictions for a lot of different scenarios. You might have heard of deep learning, which has caused a buzz recently because it can now reach human-level accuracy on tasks like image classification or NLP. So deep learning is a very powerful subtype of machine learning. The drawback of current deep learning models is that they require a lot of data to train. Now, for problems like automated language translation, this is fine, since people generate data on their phones and internet all the time. But in biomedicine, data is much more expensive and scarce. Biomedical data, as you all know, is generated in lab experiments or in a hospital, for instance, through clinical trials, which are much more costly. So by studying relevant problems in biomedicine with less data, We are also pushing the boundaries of machine learning capabilities. And with less data, statistics becomes more important. Statistics tries to understand what conclusions can be inferred from the available data. So in our work, we utilize the combination of statistics and random forests. Random forests are a machine learning method that requires less data than deep learning. I expect that we'll see much more progress on the interface of machine learning and statistics over the next years that could make AI far more capable.
which is a fascinating development. Now, we found your work, speaking of fascinating, to be fascinating. Yeah, a way to use artificial intelligence to train, to find or predict side effects of medicine, a way to offer insights into how the human body responds to drug compounds at the molecular level in both desire and unintended ways. How did the idea start for this study or initiative?
When we started this research project during the hackathon, my team member Dr. Seda Arad and I thought of random forests as a machine learning method to predict side effects from the large Novartis in vitro dataset that Dr. Urban had made available to us. Can you imagine, like, in vitro pharmacology data for over 2,000 drugs? This is a valuable resource that enabled us to pursue this machine learning approach. 2,000 drugs is really a lot, but to train machine learning models, this is just about enough for random forests. I realized that if we had a trained random forest model, I could develop the statistical methodology to identify which protein targets from the in vitro assays are associated with a given adverse drug reaction.
Wow.
So we would learn not only to predict side effects for drugs, but also which protein targets could be involved with these side effects. But in order to train this random forest model, we had to retrieve data on which adverse events are actually reported among the patient population across the US and globally for each of those 2,000 drugs. This is where Saman, with his software engineering background, stepped in. He coded the script that retrieved this data from an FDA database called FAIRS. FAIRS stands for the FDA Adverse Event Reporting Systems. We downloaded as many as over 600,000 physician reports from FAIRS so that we could utilize this information to train our machine learning model.
Wow. Now, we understand machine learning is not a silver bullet for drug discovery, but as you declared, it can certainly accelerate at least some segments in the lengthy, expensive, and risky drug development process. Can you share more about where the greatest value is in the algorithm developed?
Yeah, our algorithm helps to predict adverse drug reactions, and it adds the most value to the preclinical drug safety process. When there's a promising drug candidate at the preclinical stage, one could perform the in vitro pharmacology experiments to generate its target binding profile. This profile can then be used as an input to our random forest models, which have already been trained on the 2,000 or so marketed drugs. The model prediction will then be the risk that this compound could cause a particular ADR. And besides drug safety, our protein target ADR associations also provide insight into human biology, which could inform future basic science studies. Of course, we need to consider the limitations of our models as well. We'll discuss these in more detail later in the session, but the two major limitations are that there are several ADRs that a model cannot accurately predict. And lastly, our in vitro assays only consider a small fraction of all proteins in our body, so we might miss out on important players.
Now, you mentioned that the algorithm can spot or find trouble. Can you elaborate on that?
Yeah. I just meant to say here that our algorithm is tailored to drug safety early on in the drug discovery and development process. If you think of a hypothetical scenario where our model would predict a high risk for a severe ADR, for instance, a cardiac disorder, which could be lethal, And perhaps this could make the drug researchers think twice before proceeding with testing the compounds directly in human trials. Of course, this is a very simplified scenario. In practice, the decision process to move forward with a compound into clinical trials is much more involved. It depends on many lines of evidence. Our tool would only be one of them. And besides, other lines of evidence that are really indispensable are animal in vivo studies and other in vitro experiments.
So, we know that over the years, researchers have applied many approaches to avoid or minimize adverse drug reactions. But as a single drug often interacts with multiple proteins in the body, not always limited to intentional targets, it's often difficult to predict what, if any, side effects a medicine can generate. Moreover, if a drug does end up triggering an adverse reaction, it's often difficult to identify in which of its protein targets are responsible. Could you elaborate on this challenge?
The physiological response of the human body to a drug treatment is very complex. Let's think of this with a common drug like acetaminophen, also known as Tylenol. This is a drug against the cold. First, the drug enters the body and then the cells in various organs, such as the liver. Once the compound has entered the cell, it might interact with many different proteins. The human genome encodes for about 20,000 proteins, so the range of possibilities is very large. Through its interactions with one or more of those proteins, a cascade of events happens, resulting in a pain relief for Tylenol, but also perhaps some side effects, for instance, like headache. The question we are trying to address is which of those proteins that Tylenol interacts with sets this cascade in motion that then ultimately causes the side effect. This question is, to our knowledge, by no means addressed, but we think it is important to find out because this knowledge could help prioritize future drug candidates that would not target that particular protein that is linked to the side effect. Prioritizing such a drug candidate might thus be safer for the patient. The target ADR associations that we found are a first step towards such an understanding. That said, mechanistic experimental validation studies would certainly be needed to decisively provide any definitive evidence to show that the in vivo targeting of a particular protein would really cause an adverse reaction in the human body.
Right. Now we are aware that study researchers took one existing database of reported adverse drug reactions and another database of 184 proteins that specific drugs are known to often interact with. And then they constructed a computer algorithm to connect the dots. Learning from the data, the algorithm unearthed 221 associations between individual proteins and specific adverse drug reactions. Some were known and some were new. Could you share with the audience what the team did in the study involving Novartis and the FDA data?
Yeah, absolutely. I'm happy to dive a little deeper into the technical details of our machine learning approach. We started off with the in vitro binding assay database from Novartis, right? For over 2,000 marketed and withdrawn drugs. These are the training examples. What that means is that for a particular drug, for instance, Tylenol, its binding profile to the 184 proteins provides the input information to the random forest model. On the other hand, the adverse events report from the FDA database provided us the information on which ADRs were associated with Tylenol. Whether Tylenol would cause a particular ADR, say headache, would then be the prediction of the random forest model. Because we know the truth from the FDA database, this enables our model to learn. Formally speaking, this model is a binary classifier, since it predicts yes or no for headache. After training, we performed several different validation analyses to ensure that our model predictions were solid. We did cross-validation of drug ADR predictions. That means we compared our predictions from the model to a held-out test set where we knew the ground truth. We also performed chronological validation of drug ADR predictions by comparing our model predictions to adverse events that were reported later in time. We also performed chronological validation of drug ADR predictions by comparing our model predictions to adverse events that were reported later in time. Furthermore, we made a comparison of drug ADR predictions with drug labels from the CIDR database. And we benchmarked our predictions on OMOP, a golden standard ground truth curated by experts in the field of 400 drug ADR pairs. The next part of the study was devoted to identifying the 221 target ADR associations. Here we used Gini importance scores resulting from the random forest model combined with statistics to understand which protein target assays are important in predicting specific ADRs. We term these target ADR associations. Again, we did a validation analysis to show that our model has predictive power. To validate whether our target ADR associations predictions hold any truth, we systematically analyzed textual co-occurrences of these target ADR pairs in scientific literature abstracts. We found significantly more previous co-occurrences for our associations than for randomly selected target ADR pairs. I can give you some examples of target ADR associations that we found. The association of hERG binding with cardiac arrhythmias was one to be expected. It is well known that drugs binding to the protein hERG, which is a potassium and an ion channel active in the heart, can cause cardiac arrhythmias. And cardiac arrhythmias can be lethal. That is why drug hunters often test a drug candidate for hERG binding already at the preclinical stage. If the candidate binds hERG, that is a reason not to proceed with development of that compound. But we also found unexpected associations. Surprisingly, phosphodiesterase at PDE3 is associated with as many as 40 ADRs. PDE3 inhibitors are common anti-clotting treatments for acute heart failure, stroke prevention, and a heart attack complication known as cardiogenic shock. Doctors and researchers have known for years that PDE3 inhibitors can cause arrhythmias, low platelet counts, and elevated levels of enzymes called transaminases, a possible indication of liver damage. But it wasn't known that targeting PDE3 might raise the risk of so many other side effects, including some related to the muscles, bones, connective tissue, kidneys, urinary tract, and the ear. Lastly, another interesting association we found was for the protein BSEP, which stands for bile salt export pump. We found it was associated with 7 ADRs, for instance, urolithiasis and thyroid gland disorders, besides the well-established hepatic disorders. BSEP is mostly active in the liver, where it closely controls bile acid concentrations. There turned out to be some prior evidence in the literature to suggest that changes in bile acid concentrations might be responsible for these ADRs outside of the liver. So here we find associations of BSEP and the ADRs that might actually be indirect effects mediated through bile acid metabolism. Yeah.
Now, the associations indicated which proteins likely represent drug targets that contribute to the particular side effect and which others may be innocent bystanders. Can you explain more as to how this works and how accurate were the findings and how prime time is this tool?
Yeah, previous studies have mostly focused on identification of adverse drug reactions from post-marketing adverse event reports in patients. This has been very important and insightful, but the novelty of our approach is that we also incorporate the target-based in vitro pharmacology. This allows us to predict side effects from preclinical data, something that has not been achieved before to our knowledge. Because we have utilized target assays as features in our random forest model, we could use our trained model to identify statistical associations between certain protein targets and the adverse drug reactions. So in this way, when a drug binds 10 proteins, say, but only one of these 10 is associated with an ADR, then the other 9 will be called the innocent bystanders. It is important to note in this way, when a drug binds 10 proteins, but only one of these 10 is associated with an ADR, then the other 9 will be called the innocent bystanders. This is important to note that these target ADR associations could point in the right direction, but they remain statistical associations. I like to make the analogy with genome-wide association studies, or GWAS. GWAS finds statistical association from human population data between a quantitative trait, think like body height, and the genetic locus. Instead of a quantitative trait and genes, we identify ADR associations with proteins. In this way, there is a similar linkage between human in vivo phenotypes to the molecular players. Likewise for GWAS, further experimental validation studies would certainly be needed to decisively show any causal relation. As I mentioned before, we performed a systematic literature search for previous evidence on our target ADR predictions. But that said, if there's no evidence on a particular target ADR pair yet, it remains unclear if it was a false prediction or a truly novel finding. So more experimental follow-up studies are needed to find out. But at least these 221 associations can guide us on what to look at. So the use of our machine learning model here is to advise researchers what experiment to go after next. And all of this is done in a statistically robust and data-driven manner. I want to emphasize that our machine learning model has limitations. Targets in the in vitro pharmacology panel cover only about 1% of the biological target space, and not all drugs were tested in all assays, so our predictions are not complete. Furthermore, our predictive power for some ADRs is very limited. A good model performance correlates with the amount of reported drugs that cause ADRs. This makes sense intuitively. If no drug is ever reported to cause a certain ADR, then our model will not be able to learn anything. Also, in vitro potency, which we used as input to the model, is an initial marker of clinical effect, and it does not take into account prolonged dosing, comorbidity, or pharmacokinetic or pharmacodynamic relationships, such as the therapeutic window. And we base our model on the FAIRS data, which, although a valuable resource, has limitations in itself. For instance, the FAIRS database does not contain information on the total number of patients exposed to a particular drug. So in conclusion, it is important to consider these limitations when utilizing our AI tool.
Now, would you say the tool is ready to use in a clinical setting, or is more research and validation required?
Yeah, since I'm not an expert in clinical trials, I've consulted with Dr. Urban from Novartis on this question. We think this tool is better applied for preclinical drug discovery. Before using it in a clinical trial, it would be important to demonstrate the efficacy of our tool in a large-scale validation study. Since we have not tested this tool in a clinical setting yet, we would caution against over-interpretation of its predictions on its own. It's important to note that in a clinical setting, a large arsenal of tools for safety assessment are being used, including in vitro and in vivo animal studies, besides the human observations. We would suggest that people try it on a known case and see what our tool predicts and what not. The best way would be to compare compounds from the same structural class with diverse applications, or within the same indication, but with a varying analyte-specific reagent profile.
So how far out do you think we are from being able to use a tool like this to reduce the risk in clinical trials and minimize risks for patients if drugs gain FDA approval and enters clinical trials?
Yeah, this is a pretty difficult question that I do not have an accurate answer to, unfortunately. I hope that our tool can have an immediate impact to accelerate preclinical drug safety research. But for the clinical setting, the performance of our tool would first need to be validated in a further study. This could take several years. But I do think you raised an important point here. Like, I think that the public and the FDA would benefit from transparent regulation on how to verify the efficacy of AI tools like ours in a clinical setting. Just like there's a regulatory framework for how to demonstrate the abilities and positive effects of diagnostics. Currently, it's still fairly early days for AI in biomedicine, but how to set up a regulatory framework is definitely a discussion that needs to be held to ensure we can trust future AI tools in the clinic and demonstrate how they would minimize risks for patients.
So, what do you think this class of disruptive new technology is best suited for?
In general, I think machine learning holds the promise to add value or accelerate many different aspects of drug discovery and development. From basic science research to preclinical drug discovery, clinical trial, and post-marketing pharmacovigilance. We have seen some notable recent success stories in drug discovery, where deep learning based on chemical structures was used for de novo small molecule drug design of kinase inhibitors and antibiotics. In these basic science studies, the vast variety of possible chemical structures could be leveraged to apply deep learning. Besides efficacy, previous studies have also focused on predicting, for instance, target binding or absorption, distribution, metabolism, and excretion, so-called ADME properties. In a clinical setting, I can see how machine learning could be useful for drug repurposing and patient recruitment. And we have discussed our contribution to drug safety in length today, so there are enough case studies published now to take machine learning in the pharmaceutical industry seriously.
Well, then let me also ask you then, are there any commercialization plans?
Yeah, currently we do not have any commercialization or licensing plans. Our code is publicly available and we encourage researchers and clinicians to build upon our work. Every large pharma company has safety and toxicology experts, and we hope that our tools can help them further in their safety assessment of novel compounds that they might be developing to treat diseases, in particular diseases with unmet need. Furthermore, we have had interest already from people with various backgrounds, ranging from clinicians in hospitals to students in statistics who were keen to try out the tool. In the spirit of open and accessible science, we have made our results freely available to the public. So we hope this research can help people further.
Well, that's noble of you guys, and I'm telling you, Robert, we really appreciate your time, and we're looking forward to see how this project continues to go forward into the future.
Thank you very much, Adrian. It was a pleasure to meet you. And
You
thank you
too,
for
and
this invitation.
thanks again for stopping by.
Awesome. Have a nice day.